Hello, welcome to Biology, the scientific study of life. Active and passive immunization. Immunity conferred by natural exposure to an infectious agent is called active immunity because it depends on the action of a person's own lymphocytes and the resulting memory cells specific for the invading pathogen. Active immunity also can develop following immunization, often called vaccination, from the Latin vacca, cow. The first vaccine consisted of the virus causing cowpox, a mild disease is usually seen in cows, but also occasionally in humans. In the late 17, the English physician Edward Jenner observed that milkmaids who previously had contracted cowpox were resistant to subsequent smallpox infection, a disfiguring and potentially fatal disease. In 1796, in his now famous experiment, Jenner scratched a farm boy with a needle-bearing virus containing fluid from a sore on a milkmaid who had cowpox. When the boy was later exposed to the smallpox virus, he did not become sick. Cowpox virus protects against smallpox virus because the two viruses are so similar that the immune system cannot distinguish them. Vaccination with the cowpox virus sensitizes the immune system so that it reacts vigorously if later exposed to cowpox virus or, more importantly, smallpox virus. Modern vaccines include inactivated bacterial toxins, killed microbes, parts of microbes, viable but weakened microbes that generally do not cause illness, and even genes encoding microbial proteins. All these agents induce an immediate immune response and long-lasting immunological memory thanks to memory cells. Vaccinated persons who encounters the actual pathogen from which the vaccines was derived will have the same quick second response as a person previously infected with the pathogen. A worldwide vaccination campaign um, led to eradication of smallpox in the late 1970s. Routine active immunization of infants and children has dramatically reduced the incidence of other infectious diseases such as polio, measles, and whooping cough. In developing countries, unfortunately, not all infectious agents are easily managed by vaccination. For example, the emergence of new strains of pathogens with slightly altered surface antigens complicates development of vaccines against some microbes such as the parasites that causes malaria. Immunity can also be conferred by transferring antibodies from an individual who is immune to a particular infectious agent to someone who is not. This is called passive immunity because it does not result from the action of the recipients or on B and T cells. Instead, the transferred antibodies are biased to immediately help destroy any microbes for which they are specific. Passive immunity provides immediate protection, but it persists only as long as the transferred antibodies last a few weeks or a few months. Passive immunization occurs naturally when IgG antibodies of pregnant women cross the placenta to her fetus. In addition, IgA antibodies are passed from a mother to her nursing infant in breast milk. These help protect the infant from infection while the baby's own immunity system is maturing. And artificial passive immunization antibodies from an immune animal are injected from a non-immune animal. For example, a person bitten by a rabid animal may be given antibodies rabies. This measure is important because rabies may progress slow. To save the victim, most people who have been exposed to the rabies virus are both passively and actively immunized. The injected antibodies help keep the is the victim's own immune response induced by active immunization and by the infection itself takes over. The immune system's ability to distinguish self from non-self limits tissue transplantation. In addition to distinguishing between the body's own cells and invading pathogens, the immune system can wage war against cells and the immune system can wage war against cells from other individuals. For example, skin transplanted from one person to a genetically non-identical person will look healthy for a week or so, but will then be destroyed, rejected by the recipient's immune response. Interestingly, however, a pregnant woman does not reject her fetus as non-self tissue. Apparently, the structure of the placenta. Keep in mind that the body's hostile reaction to an incompatible blood transfusion or transplant of other tissues or whole organs is not a disorder of the immunity system, but the normal reaction of a healthy immune system exposed to foreign antigens. Blood groups and transfusions. Recall that type A red blood cells have antigen molecules on their surface. The A antigen may be recognized as foreign if placed in the body of another person. Similarly, the B antigen is found on type B red blood cells. Both A and B antigens are found on type AB red blood cells, and neither antigen is found on type O red blood cells. Individuals with type A blood do not, of course, produce antibodies against the A antigen because they are self-tolerant. However, these individuals do have circulating antibodies against the B antigen, even if they have never been exposed to type B blood. You may find it odd that antibodies to foreign blood group antigens exist in the body even in the absence of exposure to foreign blood cells. The explanation is that these antibodies rise in response to normal bacterial inhabitants of the body that have epitopes very similar to blood group antigens. For example, a person with the type A blood makes antibodies against B-like bacterial epitopes, which the immune system considers foreign but does not make antibodies against A-like bacterial epitopes, which the immune system recognizes as self. The pre-existing B antibodies in a person with type E blood will cause an immediate and devastating transfusion reaction if the person receives a transfusion of type B blood. This reaction involves lysis of the transfused red blood cells, which can lead to chills, also fever, shock, and kidney malfunction. By the same token, anti-A antibodies in the donated type B blood can act against the recipient's type A red blood cells. This later reaction can be minimized by transfusing packed cells instead of whole blood, so that donor antibodies in the fluid portion are not transferred. Notice the stable recipient's blood group, antibody in recipient's blood, and presence plus or absence minus transfusion reaction donated blood group packed cells AB, and O. Anti-B, anti-A, no anti-A, or anti-B, anti-B, anti-A. The column shaded in green indicates that a person with type O blood can safely donate blood to any recipients, and this is called a universal donor. Blood group antigens and related bacterial epitopes are polysaccharides. Such polysaccharide antigens induce immune response in which no memory cells are generated. As a result, anti-blood group antibodies are always IgM generated from primary responses rather than IgG generated from secondary responses. This is fortunate is in pregnancy because IgM does not cross the placenta, so no harm comes to fetus with the blood type that is not compatible with its mother's. However, another red blood cell antigen, the Rh factor, can cause trouble for a fetus. As a protein antigen, the Rh factor induces immune responses in which the memory cells are generated. Later exposure of these memory cells to the Rh factor leads to a production of anti-Rh antibodies that are IgG. A, protein, a potentially dangerous situation can arise when a mother who is Rh negative lacks the Rh factor carries a fetus that is Rh positive, having inherited the factor from its father. If small amounts of fetal blood cross the placenta, which may happen late in a pregnancy or during delivery, the mother mounts a humoral response against the Rh factor. The danger occurs in subsequent pregnancies with the Rh positive fetus when the mother's Rh specific memory P cells are exposed to the Rh factor from the fetus. These B cells produce anti-Rh IgG antibodies which can cross the placenta and destroy the red blood cells of the fetus. To prevent this, the mother is injected with anti-Rh antibodies around the 7th month of pregnancy and again just after delivering an Rh positive baby. She is in effect passively immunized artificially to eliminate any fetal Rh bearing red blood cells that cross the placenta before her own immune system responds to them and generates immunological memory that would endanger future Rh positive babies. Tissue and organ transplants. Major histocompatibility complex MHC molecules are responsible for stimulating the immune response that leads to rejection of tissue and organ transplants or grafts. The polymorphisms of the MHC almost guarantees that no two people except identical twins have exactly the same set of MHC molecules. Thus, a rejection reaction is mounted in the vast majority of graft and transplant recipients because at least some MHC molecules on the donated tissue are foreign to the recipient. There is no danger of rejection if the donor and recipient are identical twins or if tissue is grafted from one part of an individual's body to another part. To minimize the extent of rejection in non-identical transplants, attempts are made to use donor tissue bearing MHC molecules that match those of the recipient also closely as possible. In addition, the recipient takes medicines that suppress immune responses. However, these medicines can leave the recipient more susceptible to infections and cancer during the course of treatment. In a bone marrow transplant, the graft itself, rather than the recipient, is the source of potential immune rejection. Bone marrow transplants are used to treat leukemia and other cancers, as well as various hematological blood cell diseases. As any transplant, the MHC, the donor of the donor and recipient, are matched as closely as possible prior to receiving transplanted bone marrow, the recipient is typically treated with irradiation to eliminate his or her own bone marrow cells, including any abnormal cells. This treatment effectively obliterates the recipient's immune system, leaving little chance of graft rejection. However, the great danger is in bone marrow transplants is that lymphocytes in the donated marrow will react against the recipient. This graft versus host reaction is limited if the MHC molecules of the donor and recipient are well matched. Bone marrow donor programs around the world continually seek volunteers because of the great variability of the MHC. A diverse pool of potential donors is essential.